Okay, I think that's better. I think that'll eliminate that problem. I'm hoping that'll eliminate that problem. Okay, yeah, I did. All right, so we're going to start with touching on the eukaryotes, eukaryotes, and viruses really quickly. Just run through, remind ourselves about those. Then we're going to talk about the genetic stuff, then the metabolism stuff, and then we'll run through immunology stuff, and then try to put some of this stuff in perspective with viral, bacterial, fungal, and protozoal and helminth infections, and putting into place for the infection side as well as our response side. So to start off with, let's go over the eukaryotes. So the eukaryotes, what do we know about the eukaryotes? No true nucleus. So if they don't have a true nucleus, they also don't have any true organelles. And organelles are defined as membrane-bound structures that have specific functions in cells. And this includes things like the mitochondria, the Golgi, the ER, and the nucleus, and things like lysosomes and all that. This does not include ribosomes. Ribosomes are not organelles. Okay. They do have ribosomes. Right, so they're protein synthesis factories that are just made up of protein and RNA. So protein structures these guys have, but do they have organelles that are membrane-bound functional units in a cell? No, they don't have that. No true nucleus and that stuff. So our eukaryotes are bacteria and archaea, right? And so these guys differ based on their genetics and things like that. But for the bacteria, these are the ones that we focus the most on in our class. Remember that bacteria also have cell membranes and cell walls, and that the cell membrane and wall composition is known as the cell envelope. And then what's the cell wall made of?
I know if I saw the word on the paper, I picked the right one.
Close.
Okay.
Peptidoglycan is the wall. So gram positives, they have a thick wall. Gram negatives, they have a thin wall, but they also have something else. And what is that? Outer membrane. And that outer membrane, that has LPS, also known as lipopolysaccharide or endotoxin. All right, and like I said, these are just gonna be real basic reviews for this stuff right now, just to remind you guys about the basics for these guys. There's definitely more to these guys than just this, but I just wanted to summarize these basics quickly before we get into the genetics and the things like that, so that we can understand what's going on with these guys. So if we were to have like just a basic cell drawing of a bacteria, this might be the membrane, and it might have DNA in there, but it's not going to be contained in a nucleus. And it might have ribosomes that do the translation for us, but it's not going to be in a nucleus or in an ER or anything like that. Okay, so this might be their membrane, and then on the outside we might have a thick wall, peptidoglycan wall. Or we might have the outer membrane. Yes, so again, these guys have DNA, just to remind us. They have ribosomes, just to remind us. They will have a very thin wall. And an outer membrane. And attached to that outer membrane is where you'll have LPS. Oops. So this one will be gram positive. And again, I'll make that wall even thicker just for clarity's sake. Okay, we good on this?
Yes.
All right. Next thing was our eukaryotes. These again are cell-based, so we'll say that too. Cells. And what are cells? Do you guys remember what cells are? Just real basic. What makes a cell? What isn't a cell? So close. If it is a membrane-bound unit that is capable of making copies of itself. Doesn't matter about a nucleus. Doesn't matter. Cells. I'm not talking about eukaryotes versus eukaryotes right now. Just cells. They are membrane bound overall, and they can replicate themselves. That's a cell. Okay. So our eukaryotes, their cells, they don't have membrane bound organelles inside of the cell, but they are cells. They are membrane bound overall, and they're capable of replicating themselves. Right. So our eukaryotes, these are also cells. The difference here now, we can say these guys do have a true nucleus and the membrane bound organelles. So this would be the nucleus. What does the nucleus do? DNA. It's the control room basically of the cell. All right, then we have the, let's say, endoplasmic reticulum. Right, so this will be the protein synthesis. And other macromolecule synthesis. What's the rough ER? This has ribosomes for protein synthesis. And what's the smooth ER? No ribosomes. Site of lipid and carbohydrate synthesis. All right, the next one, we'll do the Golgi apparatus. Package and delivery, right?
That's what I was getting at.
Yeah. Exactly.
Getting the proteins to where they need to be, right?
Yeah, this is essentially, the Golgi apparatus is essentially your Amazon warehouse.
Yes.
So Amazon didn't make that crap. It's just sorting it and delivering it basically after that. So the site wherever the crap was made was in the endoplasmic reticulum, and now the Golgi is going to make sure it gets distributed throughout the cell. There might be some modification process going on there as well. So this is where package and delivery via vesicles is going to happen. All right, what's the mitochondria? Mm-hmm. Site respiration, right? ATP synthesis. Okay, so if we're going to get to the, we'll come back to this. We'll come back to this. So I'm going to highlight this one because we're going to come back to this. This is going to be important in a little bit. I don't like my choices here. All right. Let's see, lysosomes. What's a lysosome? Close, close, close. So we have digestive enzymes. We have hydrogen peroxide. We have all sorts of digestive and toxic compounds for digesting any foreign material, including bad guys. Toxins and enzymes for digestion. So we can break down whatever we brought into the cell. And so this will allow us to break down stuff from outside the cell, as well as particles that might be inside the cell that we need to recycle, that we could potentially digest it with the stuff that's in the lysosome, but that's what a lysosome will be. Is there anything else that you guys can think of that we need to go over for the cell parts of the eukaryotes? Okay, so we'll say there was one more. Chloroplast. What's a chloroplast? All right, so photosynthesis, right? So chloroplasts, we find these in plants and in algae. Algae are not plants, right? Do you guys remember the kingdom that algae belongs to? Starts with a P. Protista. So protista. So remember the protozoa and the algae fall into protista. Okay.
And it makes up 70% of our oxygen and water.
Yeah. Yes. Okay. Plants are plants, plantae, algae are protista. Protista also includes our protozoa. So let's do this then. Then we'll go through our eukaryote groups. Our eukaryote groups, to start off with in the eukaryote chapter, we first started off with the fungi. And this will include two major groups, yeasts and the fungi that have hyphae. So again, fungi are eukaryotes. They do not perform photosynthesis. No photosynthesis from these guys, right? What else do I want to say about the fungi? They have asexual spore-forming structures for reproduction. Reproduce. They also can have sexual reproduction as well. Yes, so that's the two ones, the open and the enclosed. The open ones are called conidiophores, and the enclosed ones are sporangiophores. Those are basically what we call the organisms that make the structures. The structures themselves will be conidiospores or sporangiospores. Would be the actual structures that are forming the spores, which are asexual spread of those things. It's why when you have moldy bread and you have other things that aren't touching that moldy bread, that can grow mold on it because of the spread from the spores from those asexual reproduction structures. Okay, let's move on to the protozoa. Protista, we'll do that. Protista. So the first one was the algae. These guys are basically photosynthetic, make most of the oxygen in our atmosphere. And then we have the protozoa. These are disease causing. And so we did study these a little bit more. So something about the protozoa to be aware of. They are always unicellular. Always. Okay. Another thing to be aware of with them, they have 4 motility types. Motility or non-motility. They can move by flagella, like Giardia, or Trichomonas. They can move by amoeboid motion, which we also call pseudopods. Let's write that. Pseudopod. Examples of this might be Naegleria fowleri, which causes the brain-eating amoeba infections, or acanthamoeba. Right? Then we have the ciliated guys. We did not really talk much about diseases that are caused by ciliated protozoa. There are some, but don't worry about them. We didn't talk about them in our class. And then we have the ones that are non-motile, also known as apicomplexans. And so some examples of this would be malaria. And then, yeah, I don't know. This is fine. We'll just leave it with this. So there's some examples of each of these. Okay, so those are the protista. And then we also had our helminths, which are animals. I'm going to put some dots here. We'll continue on our next page. The helminths. These are our parasitic worms. And they are in kingdom Animalia. So these guys are all going to be multicellular. These guys actually have like organ systems, like digestive tracts and things like that. They usually have complex life cycles that involve adult and juvenile stages and eggs and larvae and all sorts of stuff like that. All right. And so our categories are based as follows. We have our roundworms and our flatworms. Our roundworms are known as nematodes, nematoda. Our flatworms are known as platyhelminthes. And so we have two different kinds of flatworms as well. We have the tapeworms and we have the flukes. The tapeworms are known as cestoda and the flukes are known as trematoda. All righty. Any questions about the basics of that stuff? Yes. All right. Moving on. Then we'll go to the viruses. These are acellular. Do these guys have a nucleus? No, no nucleus, right? So we'll kind of talk about that, right? So something that all viruses have. All viruses have a genome of some kind. Now that genome could be DNA or RNA. It could be single strand or double strand for either one. And that single strand can be positive or negative sense. Right. The other thing that all viruses have. I know, that's why we're doing this. A capsid. This is a protective protein coating around the genome. It's made of subunits called capsomeres. Excellent. Some viruses might also have an envelope. What's an envelope?
So wall and a cell.
For
Membrane and a wall.
bacteria, yes. Not for viruses.
Isn't it what they take from the host cell?
Yes, it's a membrane coating that they stole from the host cell. When they're budding out. All right, so poliovirus. Poliovirus is a naked virus. Does it have an envelope membrane? Does it have a membrane at all? No membrane. It's just the capsid and the genome. That's it. Okay, so that's what we're coming back to. Coming back to these basics to remind ourselves, not all viruses even have any membrane at all. There's quite a few of them that don't have any membrane at all. They could just be capsid and genome, and that's it. Yes.
Does that make it really easy to kill though?
No, it's harder to kill the ones that don't have a membrane. And so that membrane, these guys actually rely on that membrane for their structure and their ability to merge with your cells. And because they have the membrane, that membrane is easier to destroy. Membranes are easy to destroy, basically.
It's a good target.
It's a good target.
Since that target's not there, they're hard to kill.
Yes, and so getting rid of just protein and nucleic acid is harder to do than a membrane-bound structure because a lot of our cleaning agents target that membrane. You know, when we talk about surfactants and things like that we talked about in chapter 12, those surfactants break down the membrane of enveloped viruses. The easiest of all of the pathogens to get rid of are enveloped viruses, but that just start putting it into perspective. But if I'm telling you the easiest pathogens to get rid of are our enveloped viruses, COVID is an enveloped virus. Flu is an enveloped virus. So now we see that, like, yes, they might be the easiest to get rid of with cleaning agents, but that doesn't mean that they're less transmissible or less serious of a disease or something like that, right? But that they are going to be more likely to not survive outside of the body for very long because of they have an envelope. Our non-enveloped viruses survive better outside of the body. So remember, our non-enveloped viruses are called naked. We know that. All right. So good questions. And some viruses also package special proteins with them. An example of this would be like with HIV and reverse transcriptase. And then all viruses, some other things to remember about them. They use the host cell machinery to replicate. Can't replicate by themselves. Always. And then some other things is that if they infect bacteria, we call them bacteriophages. And that if you're a bacteria, if you're a bacteriophage, you only infect one species of bacteria. Yeah, that's what we'll use for phage typing. So yeah, you guys are starting to revisit some of this old stuff, but hopefully it's starting to help you tie in some of the newer stuff and its meaning. So this was old stuff, and we use this constantly throughout the whole semester. Some of the stuff got a little bit left behind, and coming back to these details can kind of come back and tell you a little bit about what's going on with our pathogens that we've been talking about. Let's go really quickly over the lytic and lysogenic stuff for the viruses. Yeah. This is a cycle that applies to bacteriophages. Now, human viruses can do stuff like this too. We just don't call them the same term. All right. So whenever you're a virus, let me stick with our terminology for phages here. If you're a phage that is replicating and then bursting out of the cell, that's how phages get out of bacterial cells. They always burst. And this is the lytic. If the phage incorporates its genome into the bacterial genome, this makes it a prophage. And this is part of the lysogenic phase or cycle. And so lysogenic, and I remember this because of the genes, because we're incorporating in the genes. And that sometimes when you have the prophage, you can carry other genes or survival traits, which we call virulence factors. This could be toxins, could be antibiotic resistance, whatever, anything that helps with the bacteria being able to cause disease in the host. And that this process is called lysogenic conversion. All right. If this happens in humans, if you get, I'm not saying the extra gene part, but if you get a virus that incorporates its DNA into your cells, that's called a provirus. Yes, exactly. Provirus. All righty. We ready? Let's do it. Genetics. Let's start with replication. Replication is a term that's always going to be talking about DNA to DNA copies. Right? Okay, so then first we have to understand that the DNA properties. The strand is basically anti-parallel. And that what anti-parallel means is that we are going from the 5' to 3' in one direction of the strand, and the other direction is going in the opposite way. Two strands. Backbones are oriented in opposite directions. Anti-parallel. All right. And then replication, we call that semi-conservative. What does that mean? Yeah.
New strand and old strand.
Each new daughter strand consists of one old, sort of our template, plus one new that is newly synthesized from the process, right? And then if you take DNA and you are going basically from this to this, that's the idea, right? Okay, so that's semi-conservative. Whenever we're opening up DNA, though, DNA is big, long, old thing. In bacteria, they'll have one origin of replication and then can just replicate around the whole entire circular chromosome, but we have multiple chromosomes. And so we have to separate out our DNA for each chromosome. Regardless of that, DNA replication happens the same for if you're bacteria or if you're a eukaryote, it doesn't matter. And that's going to be through this semi-conservative process, and that you will have a lagging strand and a leading strand. And so what happened? So lagging strand and a leading strand. And this basically is because you will have, let's draw it out. If we're going in this way, this will be the leading, because we're able to add continuously the leading strand is always continuous. The lagging strand is made in fragments and parts. And what are those parts called? Do you guys remember? Okazaki fragments. All right, and so if this was our DNA polymerase, then what would be happening on the lagging strand is the polymerase would be able to come in 5 to 3 and make it in fragments.
And the polymerase is like the glue.
The polymerase is the enzyme that's actually adding on. So DNA polymerase is actually adding on the complementary nucleotides. Remember, A to T and C to G. So as the DNA polymerase is moving along the single strand that's opening up, it will add T where there's an A, or it will add C where there's a G, or whatever, right? Adding it on to be complementary to the other strand. So that's what the DNA polymerase is doing here. But as we're opening up this strand in that lagging strand, we're making these fragments. You can see that this is not what I wanted. This is what I want. This would be where it would be on this one. And that each of these is an Okazaki fragment. Then there would be another enzyme that would come in and connect these two. And this one is what I'm thinking you're thinking of, which is DNA ligase. DNA ligase joins the fragments. Joins those phosphodiester bonds to make sure that we're making one continuous strand. Okay, everybody following this, remembering this at all?
Mm-hmm.
Okay, any questions about DNA replication? This is the basics, right? Yes.
Just so I have it down. So the polymerase just like puts it in the right spot, and then the ligase comes and like glues it together.
Yes. Okay, we good?
Yep.
All right. Now let's do transcription. Anytime we're going from DNA to RNA, this is transcription. So for DNA, our nucleotides were A, T, C, G. For RNA, it's A, U, C, G. And so if we had a DNA sequence where we opened up our DNA,
RNA is AUCC, no?
No, AUGC.
sorry,
I've heard. But no, you still did it wrong.
What is
The
it?
G should be under the C and the C should be under,
We haven't done that yet.
oh, okay.
I'm just listing you out the
Oh.
possible nucleotides, yes. So if our DNA sequence was A, C, G, T, I don't know, T, whatever, right? Then the other side of it would be T, G, C, A. So then our RNA, we're using the T, G, C, A. Is our DNA template up top. This would be A, C, C. Yeah, I miswrote it a little bit, but that's fine. Yeah, you. But you get it. Okay. All right. Cool. Great. And so mRNA is the messenger RNA copy, functional copy of a single gene. What's rRNA? It's ribosomal RNA, part of the structure of the ribosome. Which we'll talk about in the translation part. And also important with that will be tRNA. What's tRNA? Transport or transfer RNA. And what does it do? What?
Oh, base pairs.
Amino acid. It carries amino acids based on the codon anti-codon pair up in translation. So we're going to talk about translation next, but you're not going to, transcription has no purpose without translation, right? And vice versa. So this understanding this. So these are different kinds of RNA that we get from transcription. In order to get ribosomal RNA, in order to get tRNA, we had to transcribe it from the DNA sequence first. These things don't exist separately from the DNA sequence. They exist because of the DNA sequence. So the DNA sequence also codes for these guys, okay? So these are all this stuff. And transcription is pretty straightforward. It operates very similarly to how DNA replication occurs. We're using the DNA parent strand as the template, and then we're making an mRNA complementary to it. There's 3 steps. First one is initiation. This happens at the, does anybody remember what the sequence is called where we start transcription? Promoter. It's where RNA polymerase binds. The second step, easy enough, elongation, where we will add complementary nucleotides, RNA nucleotides. And then the third step of transcription is going to be termination. Okay, that's transcription. We ready for translation?
Sure. Can I take a picture?
I mean, I'm gonna post this for you guys online. Yeah, I'll post this. I'm gonna post this on an announcement online, but I'm going through this with you guys so you guys can make your own notes as you're following along. But I'll also post this for you guys afterwards, okay? So that you have this separate. But I mean, making your own notes while you follow along is definitely better than just relying on me posting it for you. I'm sure you guys learned that by now. At least most of you guys probably have. That doing the work for yourself is what really makes the difference there. I can't tell you how many students come to me, you know, being like, oh, can I have your notes for this or that? And I'm like, make your own. like this, that, but you know, I don't have the time or whatever. It's like, I understand you don't have the time, but if you don't make your own notes, you're not
Don't go to college.
right. So I'm not saying yes, but there's work. Learning is work, right? And so you guys know that. That's why you're here, but whatever. So translation. Translation is taking the mRNA and making a protein. This is where I'll come back to this concept up here when we talked about transcription, DNA to RNA. That's how it happens in your cells. Your cells have machinery to make RNA from DNA template, right? Your cells do not have any machinery to convert RNA back into DNA unless you are infected with a virus that has its own. So HIV, even hepatitis has something like that, but they have reverse transcriptase that allows them to turn RNA into DNA. And that only works with their viral genome. They have special ability for that to work for their viral genome. It can't turn any mRNA into any DNA unless it's given certain circumstance in which that would work. And so we can do that in our genetic tools that we learned about in chapter 8. One of the things we can do is use HIV's reverse transcriptase to turn RNA back into DNA. But your cells don't have that innately, which is why whenever you get the COVID vaccine, which is an mRNA thing, you can't actually turn that mRNA into DNA. It does not change your DNA, period. Your cells don't have that ability to do that. So bringing that back around. So anyway, so mRNA, now that we've made our mRNA from our DNA copy through transcription, now we can go through translation. So what happens here is we have 2 ribosome subunits. That clamp onto the mRNA sequence. Then tRNA with an anticodon that matches the mRNA codon will attach in the ribosome and provide an associated amino acid. What that looks like is we have a big old ribosome. Big old ribosome, whatever. We have another part to it down here. And we have our mRNA. This 3-nucleotide mRNA sequence is called a codon.
And codons are only in translation, not in transcription. They're in both.
Well, they're just there in the sequence. So like the 3-nucleotide sequence in the DNA format, we would call that triplet. But once it's in the RNA format, once it's been transcribed to RNA, that same exact 3-nucleotide sequence is called a codon.
So DNA is called a triplet. And then
once it's in the RNA form, it's called a codon.
Triplet. And then exon. Are we getting there?
We'll get there. Yes. So this thing here that's the tRNA. And this part down here is the anticodon. That matches it's the complement to the codon on the mRNA. And that based on that mRNA codon sequence, that codes for a specific amino acid. And what we mean by that is that the tRNA that matches with that anticodon codon matchup calls for a specific amino acid on that tRNA. And then the next one will come in and link a new amino acid to the next guy, and so on and so forth as we move on down the line. All right, this also has 3 steps in it. Initiation, whenever we're binding. To the mRNA. And start with that start codon, which was AUG, which codes for methionine. Next is elongation again. We're going to add amino acids according to the codons on the mRNA based on that anticodon on that tRNA. And then we have finally termination when we get to a stop codon. It does not code for an amino acid. It's a nonsense codon. So that is DNA replication, transcription, and translation. Questions about the basics for this one, or any specific questions? So we had some questions on the RNA processing side of things. So with the transcription part, between transcription and translation, so I'm not necessarily going to write this down, but you guys can write this down in your notes if you want to, because we had a question about exons and introns. Actually, we'll go ahead and add this down here. Eukaryotes have this, right? So they will actually take their mRNA and it will have a series of introns, which is extra. It doesn't code for anything. We have to cut out. And exons. These are our coding regions. Spliceosomes remove introns from eukaryotic mRNA. And they join exons, so translation can occur. Okay? We good? All right. Now we're gonna take a big jump. I say big jump, couple chapters. We're gonna get to metabolism. We'll start with our enzymes. It's chapter 10, right? So like I said, we're gonna go over the hardest stuff, but I felt like reviewing the basics was important first. Now, genetics was our hard stuff too. It's been a little bit. That's our basics for our genetics. Now let's get into our metabolism. What's an enzyme? An enzyme, we can have conjugated or simple enzymes. Conjugated enzymes. What is the whole functional unit of a functional conjugated enzyme? Yeah, it's got all of its parts and it's ready to rock. We call that a holoenzyme. The protein part, where the substrate binds. Yep. The protein part where the actual active site is called the apo enzyme. All right. And then the part that is added on that helps it function, that is called a cofactor. I'll just help you with that one. All right, we have the inorganic and the organic. Inorganic, often metal ions, is just called a cofactor. But if it's organic, where you have C, H, and O, usually, typically, right? Organic molecules that are cofactors, you guys remember what they're called? Coenzymes. So a lot of our vitamins are coenzymes. Okay. They're not proteins. They're a non-protein organic molecule that helps the protein to function. That whole functional unit is called the holoenzyme. That helper part is called the cofactor. And if it's an organic molecule as a cofactor, then we call it a coenzyme. All right. Inhibition of these guys. If we have an inhibitor that binds at the active site, what's that called? What kind of inhibition is that?
That is an active inhibition. No, it's a complement.
Competition.
Competitive. If the inhibitor, so we don't know what the other one's going to be called. It's called a non-competitive inhibitor. Where does it bind if it's a non-competitive inhibitor? Allosteric. Excellent. Okay, so inhibitor binds at the allosteric or regulatory site. Basically not at the active site, but somewhere else. Then that is non-competitive. All right, so that's some basics there. Next is redox.
I'm on one sheet still. It's okay.
All right, reduction and oxidation. What is reduction?
Removal of H2O.
Hydrolysis. So you think of hydrolysis. So what's reduction and oxidation? First of all, what are these things discussing? Electrons, movements, and where they're going. So if that helps you guys, what is reduction and what is oxidation? Reduction is when you gain.
Yeah.
Right. So what do you guys use? Do you guys have an acronym that you guys remember that with? So I reminded you of a couple of acronyms that you can remember this by. Oil rig, or Leo says GER. Oxidation is loss, reduction is gain, or Leo is lose electrons oxidized, gain electrons reduced. Okay. And so in redox reactions, you always have something that is losing an electron and something that is gaining an electron. The electrons are always maintained somehow in the chemical reaction and are just moving around. I know for a fact that you learned about this in chemistry. So don't try and play with me that you didn't learn this stuff. Because as I'm sitting in on, when I sit in on Dr. Tim's lectures and I look at what you guys know when you come to my class, I
didn't
would
know
be
that.
ashamed if I were you
They
guys.
have like oxidation numbers and I was always confused on what they were. and
Yes.
how to get them.
We did have redox and Dr. Singh though.
Yes, redox. And if I were to ask you guys about your carbohydrates and how they're named and all the different structures, the Fisher models and all this
I
stuff.
can do those really well.
Yeah, I'm sure you could do it really well at the
Like the book
time.
in front
But
of
man,
me,
when people
yeah.
come to my class, that stuff has just gone somewhere else in the ether. I don't know what happens to it. Despite it being a prereq for this class, people just drop that stuff when they get here. So we could even talk about this stuff in the terms of any chemical reaction ever, pretty much. Any chemical reaction ever usually has some sort of exchanging of electrons. It might be equal and stuff like that, but there's usually almost always some sort of exchange of electrons going on in every single chemical reaction. And this is going to be particularly important when we start talking about our other stuff. So that's our summary for our redox reactions. What are the enzymes that help out with this stuff? That help out with electron exchange? So we had 6 different classes of enzymes. Let's just go over the ones that were the most important really quickly. There were 4 of them. I think they were really important. But one of them was oxidoreductases. This is specifically with electron exchange. We had transferases that literally just transfer chemical groups from one molecule to another. We had ligases. These join compounds, join molecules, they make water. And then we had hydrolysis. Hydrolases. Now, we talked about this stuff in chapter 10, but we definitely talked about this stuff again in lab. And for some reason, for some people, this crap, when we talked about it in the lab, existed separately in people's minds than what it was in the lecture. So if I ask you guys now, what is a hydrolase? See if anybody can tell me.
I think you're in water.
Yes, it does.
Usually.
Well, it uses water. It breaks up water. Uses water to break molecules apart. Not water molecules. Well, yeah, I mean, water is broken apart too, but like, if I were to actually draw this out for you in a chemical equation, what's happening is we put water into it, but the hydrogen goes onto one side of the molecule and the OH goes onto the other side. We've broken water apart, but it becomes now part of our subunits. So when you are breaking down your starch, when you are hydrolyzing starch, the enzyme amylase adds water in to basically break apart the individual sugars that make up starch. Starch is just repeating sugar units. That's all it is. And so whenever amylase breaks down starch into single units of sugar, so you can use them now for energy to make ATP, that what is happening there is that we are breaking apart water so that we can make our individual molecules again. That's what hydrolysis is. Hydrolysis is the process. Hydrolases are the enzymes. So amylase is a hydrolase, urease, a hydrolase, gelatinase, a hydrolase. They're involved in hydrolysis of those larger molecules into their smaller parts. Okay?
Mhm.
All righty. Let's move on. You know what's coming next, don't you?
No.
This one, aerobic respiration. All right, what are the 3 steps in aerobic respiration?
Krebs cycle, glycolysis.
Mhm. There we go. Exactly. Glycolysis, Krebs, and the electron transport chain. What is glycolysis?
Carbohydrate.
Glucose, a 6-carbon glucose, is being broken down into 2 3-carbon pyruvic acids or pyruvate. The goal in glycolysis is to make pyruvate. All right, so that's the goal. Do we make anything else in glycolysis? A small amount of ATP, net 2 per glucose. Do we make anything else? Does anybody remember? We also make this. Do we make carbon dioxide in glycolysis? Right, excellent. All right, Krebs cycle.
That's where you make the bulk of your.
Let's, yes, so it's not where we're gonna make our ATP. God, it. Come on, dude. Give me a break here. Okay. There's kind of an intermediate step that we have to go through before we go through Krebs. And that is taking pyruvate and turning it into acetyl-CoA. Coenzyme A. When we do this, because pyruvate's a 3-carbon compound. How many carbons are in acetyl-CoA? 2. So what happens? What do we give off? If you're losing a carbon, you got to give off CO2. Okay, so that's where our first carbon dioxide comes from. So now we put acetyl-CoA into the Krebs cycle. And we're going to turn it into basically a 6-carbon intermediate and cycle it. We're going to make out of this cycle CO2, a lot of it. We're going to make a little bit of ATP and lots, the whole goal, of electron carriers. Our electron carriers in the form of NADH and FADH2. Where are the electrons in these molecules? The hydrogens. So what we do when we're making those, they start off as just NAD, and it's going through Krebs or whatever. We're going to add an electron in the form of a hydrogen, NADH2. NADH, and it'll come with a H plus usually. The whole point of Krebs is to make our electron carriers. The whole point of glycolysis is to make pyruvate so that we can cycle pyruvate into acetyl-CoA and then push that into the Krebs cycle to make our electron carriers. Then the whole point of these two guys is to get to the electron transport chain. In the electron transport chain, we have your NADH and its H plus that comes with it. Give an electron into the electron transport chain, which is a set of proteins on our membranes this whole structure here is called ATP synthase. What's ACE mean? Enzyme. Right. So what this is going to be doing is allowing us to add a phosphate group onto ADP to turn it into ATP. So that's really what this is going to be doing. But this is all driven by hydrogen ions coming back through it. So we will take ADP and turn it into ATP, which is the goal. This is the goal of this part, right? The whole goal here is to make ATP, which is our energy guy. But in order to do that, we have electrons getting shuttled through here, through this set of proteins. Does anybody remember what these set of proteins are called? They're called cytochromes. Starting to sound familiar? Cytochrome C oxidase. This is the last one. It's this one right here. Cytochrome C oxidase. I'm not going to test you about that on the lecture exam, but now this should at least put into perspective what I was talking about with cytochrome C oxidase being important in aerobic respiration for aerobes. So what happens here is these H's, the H pluses that we're giving off, and we'll give another H plus off of this when it donates its electron. So these H pluses get shuttled out here. We'll just put pluses around. This is all H pluses. So now we have a gradient of pluses on the outside of the membrane. And this is a whole ass membrane. This whole membrane circulates the whole entire mitochondrial matrix. In the case of us, this will be happening on the membranes inside of our mitochondria. The mitochondrial cristae is what they're called. The cristae are the folds inside the mitochondria. This happens on the cristae in the mitochondria. In bacteria, where does this happen? Not on the mitochondria, right? The inner membrane. This has to happen on a membrane because these cytochromes have to be on a membrane. So the inner membrane of bacterial cells, this is where the aerobic respiration will occur. Any respiration, aerobic or anaerobic. If we're going through an electron transport chain, it has to happen on a membrane. So this happens on the inner membrane of the bacteria. So we've got a lot that can happen on or inside of the bacterial membranes. So if you're going through transcription and translation in bacteria, we know in eukaryotes that you will have your DNA inside of your nucleus. So in order to make your RNA, that has to happen. Transcription has to happen in the nucleus. Then we move the mRNA out of the nucleus into the endoplasmic reticulum, or the ribosomes will translate it. In bacteria, we don't have a nucleus. So it all just happens in the cytoplasm. But when we start talking about making our energy, our electron transport chain happens on a membrane, period. So in order to make that energy in bacteria, they do this on their inner membrane, just that inside membrane, inside of their cell wall. This is going to be happening. So we'll move our electrons down through the cytochromes. And as that's happening, the negative electrons interacting with each cytochrome, that causes protons to be pumped outside. Now our protons will want to move inside. So we'll get our H pluses back inside, and that powers ATP synthase to add a phosphorus group onto ADP and turn it into ATP. This electron, after it's moved through all of the cytochromes, we can take, that is not a plus, we can take that electron minus and the hydrogens and bring in O2 and split it up. That the electrons plus the H plus splitting up an O2 will give us H2O as a product. And this is why is this doing this? This is why oxygen is our final electron acceptor in our process. Okay. If I were an anaerobe, a true anaerobe, what's different?
No oxygen.
No oxygen. So they'll use nitrate or sulfur products or something like that. And so when we talked about that nitrate test that we did, the nitrate reduction test, they might be using nitrate here instead of oxygen. If we talk about sulfur reduction, they might be using a sulfur or sulfate product here instead. And then they're making H2S instead of H2O. That H2S that we tested for with our iron products in SIM or TSIA in the lab. Remember that? All right, that's what's actually happening, and that's where it's happening, is in that. For true anaerobes that can actually go through anaerobic full respiration. There's another type of anaerobe called a facultative anaerobe. What do facultative anaerobes do?
They ferment.
They can
They
switch.
ferment. They can switch between aerobic respiration when they have oxygen, and then fermentation, we just get stuck in glycolysis. So they will in glycolysis make that little bit of ATP right here in fermentation, and that's the only ATP they make.
So how much energy does fermentation make versus aerobic respiration?
So fermentation will yield 2 ATP for every single glucose molecule that goes through glycolysis. Whereas if you're an aerobic respirator and you have oxygen available to you, you'll make 38 ATP for 1 glucose molecule. So very inefficient to go through the fermentation process, but it's better to get some ATP than none. And it's so imperative that we keep up our ATP in our body that when I suffocate you and you die, it's because you ran out of ATP. We think, oh, because you don't have oxygen. What the F do you think you need oxygen for? To make ATP.
But that's rigor mortis.
You run out of ATP. Yes.
You no longer have ATP. That's why you're.
Yep.
That's why you die.
Yep. Okay. We good?
Very-
Sure. Any questions about this stuff? Okay, so again, we're going through the basics of this stuff so we can at least like reorient ourselves with those concepts there. So that's that for that one. And then our next last big hurrah is immunology. All right.
Yay.
So let's start with our innate. We've got like 10 minutes left, but we'll try to crank out what we can. Let's just do, yes, inflammation is for sure. Let's just do it like this. Let's just talk about the immunology of it. Let's not try to divide it up as one or the other. So what are your white blood cells known as? Leukocytes.
You have agranulocytes, granulocytes.
Mhm. So we have the granulocytes and the agranulocytes. What are your granulocytes?
All my fills.
The fills and one other thing, mast cells. All right, what are your fills? We got 3 of them. Neutro, eosinophils, and then basophils.
I remember it's big. Being this is brain books.
All right, and then what are your mast cells? What process did we talk about mast cells in?
Tissues. I say tissues, no?
Our adrenal.
Right, remember the mast cells wear the IgE as like a surface receptor, and whatever IgE you have against whatever you're allergic to, whatever gave me my rash on my neck, probably this is probably contact dermatitis, which is probably delayed type response, which is my CD8 T cell response. It's probably some necklace or some crap that I wore 2 days ago that I'm now getting this horrible blistering rash from, but it's also probably got some IgE in there as well. It's usually got both going on, but it is what it is. All right, so agranulocytes. We had our lymphocytes,
These are monocytes.
and then our monocytes. What are our types of lymphocytes? B and T cells. And then our monocytes got broken up into macrophages and dendritic cells. All righty. Great. All right. So neutrophils. So let's talk about phagocytosis. No, that's not how you spell this. Phagocytes. Which cells are our phagocytes? Our major ones. Neutrophils and our monocytes, including our macrophages and dendritic cells. All right. What about our antigen presenting cells? Yep. Yes. So these are the cells that have MHC2. We're right about this. We're not wrong about this. So which cells can have that?
So, and
Wait.
all cells have MHC1.
Mm-hmm.
Everybody can
Because
call
they're all
911.
Mm-hmm.
And antigen presenting cells are your macrophages can present, your dendrites can prevent, and maybe neutrophils.
All right. And then B cells.
B cells, I always forget those guys.
Yeah. These guys are not phagocytes. So if B cells were going to act as antigen presenting cells, where did they get the antigen from?
A B cell is an antibody, so it's a reaction to an antigen.
Sure. So B cells can see antigen without having been activated first. They can't make antibody until they're activated by a captain. But how do they interact with their antigen before they make antibody? How are they interacting with the antigen that activates them? They have a receptor on their surface.
C1.
MHC does not recognize specific antigen. It can present any antigen, very non-specifically. So there's a receptor on T cells that recognizes specific antigen, specific to that B cell.
So the T cell is what?
It is called IgD, y'all. IgD is the receptor on B cells. Ah, okay. IgD. So it's like a membrane-bound version of the antibody. And it's a receptor for that B cell. So what happens is that B cell's just waiting in your lymph node with that IgD on it. And that IgD sees the antigen that's coming through your lymph node. That could be the bacteria with the antigen on it, or just part of the bacteria or virus or whatever it is. Comes through your lymph node, that B cell with its IgD, the IgD sees the antigen, brings it inside the cell and presents it on MHC2. And now, what is MHC for? What does MHC do?
Major histocompatibility complex.
All right.
Gene for cell markers.
Major histocompatibility complex. Okay, so major histocompatibility complex. That's what MHC is. What does it do?
Coding genes, coding genes for cell markers.
Now, what does it do though? What's its function in immunology?
It's for the inside, right?
Don't give me a definition. Tell me what MHC does.
Self-recognition by immune system and rejection of competition.
Don't give me a definition.
It's like
Explain it to me. Kind of. That's part of what it does. What does MHC? It can do that, right? So my body has MHC, Lauren MHC, and then Haley's body has Haley MHC, and my body sees that as foreign. Sure, but what does MHC do in the immune response? What's its normal function? Kind of, yes. But what is MHC's function?
It tells me who's me.
Kind of. No. Yes, it does do that, but that's not its function. That's like a secondary thing. What's its actual function in the immune response, guys?
To not overreact.
Finding if there's an infection inside a cell.
Kind of. We're not getting at it. MHC just presents antigen to C cells. That's what MHC does. The function of MHC to present antigen to T cells. That's really what it does. That's its function.
T cells are the
So why? So we haven't gotten that far, right? So we're just saying right now, MHC, we got to break it down first. MHC, no matter what kind of MHC, always, MHC presents antigen to T cells, period. There's two types of MHC, MHC1 and MHC2. MHC1 is a marker that presents antigen to T cells, but more specifically, it is a marker that presents antigen to CD8 T cells. Whereas MHC2 is a marker that presents antigen to CD4 positive T cells. All right, so here's some characteristics we can use to compare these guys. What type of antigen does MHC1 present? Inside or endogenous antigen, whereas MHC2 presents outside exogenous. All right, so MHC1 always presents inside antigen, and MHC1 presents it only to CD8 positive T cells. CD8 is the marker on the CD8 positive T cell that recognizes MHC1 is presenting it. Okay. CD4 is the marker on CD4 positive T cells that recognizes that MHC2 is presenting it. All right. So this is MHC and its function. MHC1 is on all nucleated cells. MHC2 is only on antigen presenting cells. And this is how T cells interact with antigen. So let's move on to that then. T cells, they only react to antigen presented on MHC by a presenting cell of some kind. All right, the T cell receptor, also known as the TCR, recognizes the specific antigen. So what happens is you have your infected or presenting cell. So let's start with our antigen presenting cell. This is our macrophage. And it found some antigen outside. It phagocytosed it. And then put it onto MHC2, presents the antigen. Then my CD4 positive. This is on MHC2, so it has to be a CD4 positive cell, right? T cell, also known as what kind of cell is CD4 positive? T helper.
No, one. No.
Just T helper. Just T helper. Yes. So good. We're talking about it. So this is the T cell receptor, recognizes the antigen. And then what marker recognizes the MHC2? CD4. So this is what's actually happening. All right. So that antigen presenting cell got outside antigen, put it onto MHC2, which presents outside antigen. The only type of cell that can react to MHC2 with antigen is going to be our CD4 positive T helper cell. Now that T helper cell gets activated, they cross-activate one another because of this interaction. And then that leads to TH1 and TH2 always. Always both of them will get made. Now, TH1. This one will be activating our infected self cell. It's infected on the inside with antigen. This is inside, so what MHC does it present on? MHC1 presents the antigen. And then you have a whole other ass T cell with a T cell receptor. I didn't mean to zoom in on that. Yes, thank you. So this is, so what's CD8 doing here? CD8 recognizes MHC1. Always. This is our T cell receptor that recognizes the specific antigen. This is our CD8 positive cytotoxic.
It's just gonna kill it. No questions asked.
Right, but it can't do that without the signal from our T helper cell.
MHC, that's your common. Okay. So then the cytotoxic cell is like the ambulance or the police. They just go. They don't care.
No, they don't go without the go-ahead from the captain.
Well, you have the call, and so that's your 911 operator.
So your call is your MHC1, I guess, technically. And then your 911 operator, I don't know how you want to integrate that. It could be your TH1. So the 911 operator is kind of signaling to let the go-ahead happen. But you being infected on the inside, your call out for help is your MHC1.
That's 911.
Yeah, that's your 911. And so the CD8 T cytotoxic T cell will come and respond to that, but it cannot do shit without the go ahead
Right,
from
but
the
when you-
TH1
But
cell.
when you call 911, you're calling the operator and the operator
Sure.
dispatches
So if you want to
the
see the,
policeman.
right, and so if you want to see the
And he just
TH1,
comes and takes it out.
yeah, so if you want to see the TH1 is that operator, but the T helper cells are our captain cells, right? And so the captain is what gives the go ahead to do that. In your actual B cell response, your B cells won't make, it's the same kind of thing, but we'll be presenting on MHC2, right? We'll activate our T helper 2 cells just before you guys go and head out because we got to go. I have another class at 1, but the T helper 2 cells, the TH1 cells activate the inside response. And the TH2 cells activate the outside or the antibody. So then you would have a B cell that activated with antigen.
See, I just feel like this is 911, this is the operator, and that's the police.
Whatever works for you. I will say this, the IgD receptor sees the antigen. Oh, come on, stop doing that. It comes inside, it gets presented on MHC2.
We're doing this Wednesday too, right?
No. So Wednesday is just going to be the meta discussion. So I'll expect you guys to already have this under your belt on Wednesday. Yes. So that's why I wanted to kind of go over this first. But really quick, our T cell response, this is our CD4 positive T helper. And we'll say this is a TH2 that already got activated from above. And then TH2 activates the B cell. And then this becomes a plasma cell. That plasma cell, what does it do? Makes the antibody. It also makes it in memory also. So the plasma cell starts out first by making IgM, and then we'll switch to IgG, IgA, or IgE. Yep. Yeah, and it's the membrane-bound version. It's the receptor on the B cells. Right? And so that's IgD. And so then IgM is our first antibody. It's the pentamer. It has some basic functions, but IgG is our workhorse in the bloodstream. And it is also going to be important in memory. It's the fastest and the strongest. IgA is our dimer. It is the secretory antibody. It's found in the mucosa. Mm-hmm. And then IgE is associated with either allergy or parasitic response. So this will be our protozoa and our worms. Yeah. Okay. You guys have the gist of this down? I know that this this MHC stuff, that's the hardest stuff. So if you guys are trying to figure this out, I hope that this sort of like helped you guys figure out. MHC presents the antigen to the T cells, period. That's what it does. And then MHC1 presents inside antigen to CD8, and MHC2 presents outside antigen to the CD4.
And exams Monday at 10 in the lecture room.
Yes. All right. And so we'll be back on Wednesday to do our meta discussions where I'll be asking you more like big thinking, like incorporate all of our things.
So we have lecture and then
Yes, on Wednesday. Yes. So lecture will be our presentations, our unit 4 presentations. Yes. Any other questions, guys?
I redid mine. I don't know. You said put it in the comments, so I put it in the comments, but I also did resubmit.
Yeah.
So
Yeah. Should be good.
I'll send you mine probably later today.
Okay. Yeah.
Can I do that? Just send it to you?
Yeah, if you want to just email it to me, I'll take a look at it. And then I'll email you back if I want any corrections or anything. Yeah,
Okay, awesome.
Yeah.
Thank you.
I'll save you the time. You'll see this and then my online people will actually show up.
thank you.
Yeah.